One important lesson that the coronavirus pandemic has taught us is that we, as phylogeneticists, were not prepared for the volume of data. We live in the era of big data. We constantly talk and think about massive databases created in recent years and the research they support, groups like the SRA and GISAID. Despite this, we have continued to only innovate our tools as much as necessary for specific research applications at any given time. And when the COVID-19 pandemic inspired massive global sequencing of SARS-CoV-2, we were woefully underprepared. Many current tools take hours or completely fail to process a tree of only a few thousand samples, and we have been sequencing tens of thousands of virus genomes daily across the world. Our group recognized this issue early and began developing efficient software for the analysis of phylogenetic trees on the order of millions of genomes in the time frame of seconds. Our first tool, Usher, which places new samples on a large phylogenetic tree, was completed and published last year. This tool was our first step in addressing the scale of coronavirus data and can map new samples to a tree of millions in seconds. Alongside Usher, we are now also providing a daily updated repository of global phylogenetic trees through the Human Genome Browser. However, simply constructing and adding to this tree is not enough. Here, I present a way to put those massive global trees to work, matUtils. Before we proceed, it's important to describe our core data structure. We are using a mutation annotated tree, or MAT. Essentially, this is a parsimony phylogenetic tree where all mutations are noted at the node where they occurred rather than as a part of each sample as in a VCF. This provides a form of evolutionary compression and allows us to distribute a complete phylogeny and full sequences of millions of coronavirus genomes in less than a gigabyte of data. Our implementation of a MAT is stored in Google's efficient and customizable protobuf format. While incredibly powerful, this is a non-standard data format. Currently written phylogenetic tools are not capable of parsing the MAT file that Usher can construct. This is where matUtils comes in. It is a suite of tools for format conversion of MAT protobufs, selection, subsetting, and statistical calculation designed to support open and public research use of our MAT database. matUtils comes with 4 basic commands and a few more specialized options. The basic commands are summary, annotate, extract, and uncertainty. We can see these visually displayed on the right side of the poster in Figure 1, along with a 5th, somewhat experimental option that we will dive into shortly. Summary is essentially a view function that can calculate basic statistics about node and sample count in a protobuf, among other things, to help the user understand the shape of the file before performing an analysis. Extract is our workhorse function, performing all selection of samples and manipulation of the tree itself, with a profusion of options from selecting based on having a specific mutation to evaluating a regex to select samples from the tree by name, and capable of generating VCF files, NUIC files, or auspice-compatible JSON files. These JSON files can be used to generate a visual of your tree or any arbitrary part of your tree right away using the NextTrain online interface. An example of this is shown in Figure 2. Annotate is a more specialized function used for the addition, subtraction, and estimation of clade and lineage identifiers. We maintain the clades and lineages tracked by NextTrain and by the pangolin groups as a part of our provided database. Finally, uncertainty is a function for calculating sample placement uncertainty across samples that are already existing on the tree. Many times the user will want to ensure that there is strong support for a specific sample being in its location on the tree. Uncertainty can calculate whether and how many other places on the tree fit that sample equally well, as well as an accompanying metric we developed called the neighborhood size score. The neighborhood size score essentially reflects how widely distributed those multiple equally good placements are. Together, these functions form the backbone for the application of MAT protobuf files in pandemic-specific and more basic phylogenetic research. Further information on any and all of these functions can be found at our wiki page. Now we turn our attention to some of the ongoing development in our group. I've been specifically interested in heuristics for phylogeographic analysis and more generally in the manipulation and tracking of metadata on our tree. This has some useful applications bundled under matUtils introduce as we see in the last part of Figure 1. matUtils introduce can take a list of samples associated with an area or region and return a ranking list of clusters of samples grouped by inferred point of introduction or common ancestor in the region. matUtils introduced can also identify where an introduction may have come from, if multiple regions are passed and there is support for the origin ancestor having been a member of a specific different region. Introductions are inferred by heuristic calculation that we developed that is essentially a weighted summary statistic for each internal node describing the composition of descendants from that node in terms of our category of interest. This is not a full Bayesian phylogeographic analysis, but it appears to perform well and only takes a few seconds to calculate for regions of thousands of samples on a background of millions, instead of multiple weeks on a much smaller data set. We demonstrate the viability of our approach in Figure 3 on the right. In Panel A, I display the log count of introductions into the state of Pennsylvania from the rest of the continental United States where the origin was able to be inferred. This count is very biased by this level of sequencing at the origin. Naturally, it's much easier to identify an introduction from a region that is well characterized in the data than one that we do not have information about, which would simply appear as a generally indeterminate introduction. To account for this, I divided the number of actual introductions detected from each other state by the number of all introductions which arrived from anywhere to the target state, being Pennsylvania in this case, then took the log of the ratio. This is displayed in panel B. States that contribute more introductions than expected appear bright on the seed map. Note that there is no inherent geographic information included in the generation of the seed map beyond that implicit in COVID-19 population structure itself. This method is fast. I can and have generated similar statistics and plots for all 50 continental United States in a few minutes of total compute time on a single thread on a laptop. The future of this method is undecided, and I'm hoping to gather feedback and suggestions on directions we could take this type of heuristic analysis. Ultimately, this at least serves as an example of the scale and speed of analysis our database and packages support. In conclusion, here I present a new toolkit and accompanying database to support open and rapid phylogenetic analysis. We live in the era of big data, and our tool lets the average user with an average computer efficiently analyze samples in the context of every coronavirus genome ever sequenced and posted to public databases anywhere. We want to support variant of concern scanning and monitoring, contact tracing, and your research into coronavirus and more general viral evolution. I welcome question, comment, or suggestion related to the toolkit described here, including supported formats and useful summary statistics we should include to support your research. Thank you all for your time and attention.